the thank you for the kind introduction. So what I'd first like to do is, before I start, I've some slides to show, and the reason I have slides to show is because it's quite often a bit easier to explain things through pictures, and that's one of the reasons why I like, you'll see a lot of drawings and some pictures to help explain the journey. And before I start though, I'd really like to ask the group is, when you hear the word DNA, what do you think of, and what are the things that come to mind when you hear the words DNA?
Did not attend.
Fair enough. No one said that ever, so that's another one for me. So I've got in the chat, Lindsay has put genetics. So yep, that's DNA is an information material. It's our genetic material. So yeah. Anything else? Laura? Yeah, do free just to shout out. It's fine. You can unmute and shout out, but you're muted, Laura.
I'm very good at putting myself on mute. I was just raising a hand because I actually did a mini course at UCL in the 1980s in genetics.
Yeah, okay. That's pretty cool.
Yep. So when you said DNA, what do you think of? I thought of UCL.
UCL, okay. Well, I mean, I'm from there now, so yeah, hopefully we'll be able to touch on some things that might seem familiar to you. We've got criminal record in the chat as well, and that's quite often quite a lot of people think about it's DNA testing, crime scene recovery of DNA. And then Helen's suggested double helix, which is the shape of DNA that Watson and Crick first proposed in 1953. So quite a mixture of different things that we think about when we think of DNA. But what hopefully I'm going to try and explain to you is that DNA can be more than those things, and how that might relate to diabetes and what we're looking at. So what I'll do now, if those that haven't already muted don't mind muting themselves or being muted, wonderful, thank you. I'll share my screen if that works. Sometimes I don't want it to open up to my acknowledgement slide. It's a bit of a spoiler, isn't it? Go straight to the good bit.
Yes.
Right, there we go. So changing shape of DNA and diabetes. So my name's Zoe and I'm from the UCL School of Pharmacy, and I'm an associate professor in drug discovery. And what I'm going to talk to you about today is about one project that's in that we are working on within our laboratory, but largely we look at the changing shape of DNA in lots of different things, and diabetes is one of those. So we've talked about DNA, and there's lots and lots of different ways that you can think about DNA. So we covered like criminal record, so that sort of relates to crime scene investigation. I haven't had anybody shout out Jeremy Kyle for a while, and that's probably because he's come off the TV now. But paternity testing's another one. Mr. DNA from Jurassic Park. And then we've got things that are a bit more recent. So Angelina Jolie had genetic testing to determine her predisposition to certain types of cancer. So one of the things that she found out, she actually has a particular gene that means that she's really, really likely to get breast cancer and also ovarian cancer. So based on that genetic testing, she has elected to have a double mastectomy and have her ovaries removed off the basis of that genetic testing. And although it seems like quite a radical thing to do, but this is moving forwards into the future, we're more likely to do that on an individual basis. And actually you can go and have your DNA tested. There are commercial tests. So 23andMe do one. There's other different ones on the market as well that can give you your predisposition to certain types of conditions, or even actually where, you know, how much Viking are you and things like that. DNA is also involved in genetically modifying food, so trying to make plants better and crops more fruitful, but also less likely to be overcome by different types of disease and things like that. We don't hear so much about it, so it's kind of, there was quite a lot of opposition to start with, but that's also modifying DNA, and also modifying DNA in animals. So these are animals that have green fluorescent protein in for different types of experiments. So you can see that they're coloured green, and that's because they've been given a small amount of a gene to enable them to express a particular protein. So DNA is everywhere, and depending on, you know, what types of things you read and what types of media you're into, that you could have lots of different perspectives on what that is. So we're going to start with what it is in terms of humans. So in our bodies, we're made up of lots of different types of cells, and the cells make up what are our tissues. So we have different tissues for different parts of our body. We have different cells for those things. So we have bone cells, we have skin cells, blood cells, all of those different cells, many different types of cells in the body, and each of them, or most of them, have a nucleus, which is where our chromosomes are kept, and chromosomes are made up of, in part, DNA. DNA itself is made up of 4 building blocks. They're quite often referred to as the building blocks of life, and we refer to these as G, C, A, and T. And if you're interested in the chemical structures, this is the chemical structures that they look like, but we quite often, even my group, we refer to these by their letters. So we talk about G's and C's, A's and T's. And these 4 building blocks, they hang around in pairs. So G always likes to hang around with C, and A always likes to hang around with T. So they're paired for their properties, essentially, and that will come in a bit more clear, hopefully, in a couple more slides. And our genetic code is the sequence of bases that are needed to code, so the information to code into what to create that, whatever it is. So for example, the genetic code for humans is 3 billion bases, or 3 billion letters long, which is massive. And you'll see that mice have a much smaller, slightly smaller genome than us. So it's 2.75 billion letters long. Worms are smaller. E. coli is much smaller still. So E. coli is a type of bacteria, as you might know. But you might be surprised to find that newts have a much larger genome than us. So it's this we quite often think that we're the most complicated organism on this earth, but actually newts, their genetic code is much bigger than ours. And in fact, if we are going to go so far to say, well, what organism has the biggest genetic code on the planet, and actually plants have the largest genetic code. They have so the largest genetic code is 148 billion bases long, so that's much bigger than ours. So this rhododendron that I'm stood in front of in this picture actually has a genetic code that's much bigger than my own. And so what does DNA code for? So I've already said it's an information molecule, but what does that mean? What does it code for? So DNA is like a blueprint, and it's used to make a working copy of a molecule called RNA. Now, up until about 4 years ago, you probably might not have heard of RNA. You may well have heard of DNA, but perhaps not RNA. But now, because of the mRNA vaccines and the fact that COVID-19 is an RNA virus, you're probably more familiar with the term RNA. But RNA is quite similar to DNA, but it's used as a working copy. So DNA is like a blueprint. RNA is a working copy of that blueprint, and that's then used to make proteins. So, for example, in the insulin gene, the DNA is converted into RNA, which is then used to make the protein hormone insulin. And the way that I like to think about it is, if you think about DNA being an important code, you don't want to damage it. You don't want to do anything bad to it. You want to protect it at all costs, because if you make changes to the DNA, that's going to affect everything. So I like to think about it as, imagine you've got a signed copy of a really, really, good book that you like, let's say Mary Berry 100 Cakes and Bakes, and you want to make cupcakes. Now that book, imagine it's the only copy left. There's one copy, it's signed, and you still want to use it, but you want to keep it safe. So if you want to make a recipe, you'll take that recipe, then you'll make a photocopy of that recipe, or take a photograph of that recipe, and then you can then use that to then make your cupcakes. And that's essentially what the body's doing with the DNA. DNA is an information molecule, but it's not read directly. So rather than just making the cupcakes straight from that recipe in the book, we use an intermediate photocopy, working copy, then to make those whatever we want to make. And this is the insulin gene. So these are the letters, the code that comprises the insulin gene. So everything in bold here are all the letters that are needed to express, and that's the word we use, our body to make that gene. So we express genes. So gene, we call things like making a protein from a gene, we call that gene expression. So all of the stuff that is in bold is the code needed to make insulin. And we're actually largely not interested in this bit. We're quite interested in the bit in front. But why is that? I guess is the big question. But before we then delve into why we're interested in which part of the insulin gene, I'd like to talk a little bit more about DNA. We did talk about how DNA is inherited, so genetics. And I don't know whether you can recognize this, but the fact that I'm wearing a 1980s, the fact that this person here is wearing a 1980s velvet dress shows that this is me when I'm about 18 months old. And you'll see some familiar features in all of these babies here, but they might look very similar, but this is me and these are my three sons. And you'll see that we do look very, very, very similar. It is uncanny how similar I look to my children. And you'll understand that from looking around you and seeing people and seeing families that look similar to each other. And quite often people will say, your son has your eyes. And that's really sweet and actually really accurate. Like, no, we have very, very similar eyes. But what they should be saying, and what would be more accurate scientifically, is to say, your son seems to have inherited a gene or genes from you that make a protein which instructs the cells in his iris to produce eyes which are the same color as yours. Now, that doesn't quite sound the same, does it? But essentially, the reason why my sons look the same as me is because they share the same code as me. So they're using essentially the same code as I am, or I have done in my appearance. And that's how we inherit parts from a parent. So you inherit 50% of your DNA from your father, 50% of your DNA from your mother. It gets mixed up, and sometimes it gets mixed up and you won't have a condition, and sometimes it gets mixed up, and even though that neither of your parents have a particular condition, you end up having a particular condition. And that's why some people will randomly get diabetes, whereas some people, it will be in the genetics will be inherited throughout generations, and you'll have whole families that will have diabetes. Because I'm interested in the shape of DNA, it's quite important to think about what DNA looks like. So one of the things that was mentioned at the start was DNA is a double helix, and this is what a rotating double helix looks like. The reason I love this GIF is because it's quite often used in sci-fi films, and I have no idea why. It's usually in the background on a computer, and somebody thought it looked fancy, so I thought I'd keep it here. But this is the shape of DNA, and DNA could be described as being shaped like a twisted ladder. And what we mean by that is if you take a ladder and then you twist it round, you'll get this type of shape. But what do we mean by a twisted ladder, and how do the letters relate to what we see in that double helix? Well, the sides of the ladder, the handles of the ladder, are where these bases would be, and that's where these letters would be. So we've got G base pairing with C, A base pairing with T. Those form the handles of the ladder, and the rungs of the ladder, so those bits in between here, these are interactions between these bases. So these are forces that hold these bases together. So when we say that DNA is a double helix, it is because it's not just one thing. There's two things, two handles of your ladder, and that's what causes it to be a double helix. So you've got two strands of DNA, essentially. You've got one strand here and another strand here. And it's really important that there's two strands, because it means that if you want to copy DNA, you can just take those two strands apart. And because of this special pairing, like G always hangs around with C, A always hangs around with T, it means that you've got a perfect complement. So you can take those strands apart and then infill, like use one as a template to copy. And that's part of how DNA replicates, and how and that's why it's important that there's those special pairing properties. So we've known it's a double helix since the mid 1950s, and Watson and Crick were the first scientists to propose the structure of DNA to be a double helix. And quite often they are attributed to discovering DNA, but that DNA was discovered by a scientist called Friedrich Miescher in the 1800s. He was a very quite a peculiar person who liked to collect bandages from his local infirmary and extract the pus out of them, and then from that pus he actually found what he called nuclein, and that's essentially he was extracting DNA out of these bandages. So it was Friedrich Miescher that discovered DNA. Watson and Crick were the people that first proposed the structure that ended up actually being correct. And it wasn't until the 1980s that it was proved by X-ray crystallography that this was the actual structure of DNA. So this is what we mean by a double helix. This is another way of depicting it. We thought, well, actually, is that it? Is that the end of the story? Well, if it was the end of the story, I wouldn't be here. So in 1962, they showed that actually, if there was a lot of the base G, that the DNA could fold into some really strange shapes. So rather than it being a double helix like this, it could actually fold into almost like a cube-like structure. And then in 1993, it was shown that if you had a lot of the base C, you could form this very tightly compact structure, which is, if you interlock your fingers, the core of this structure is almost like that. So rather than it being like a ladder, if you interlock your fingers, that structure was similar to that. And in 2003, they completed the Human Genome Project. So this project was started in the 1980s. The idea was to determine the exact sequence needed to make a human. And so determining how big the human genome is. So that's how they found out it was 3 billion base pairs. But something they also found out is that out of those 3 billion base pairs, only 1 to 2% of it actually codes for protein. And then the next question was, well, what on earth does the rest do? So out of those 3 billion base pairs, if only 1% coded for protein, why do you need the rest of it? But it was apparent through people that had lost bits of DNA and different experiments showing that even though that DNA wasn't used for making protein, it wasn't needed to code for protein or different things like that, it was critical. If you lost it, it was disastrous consequences. So it's initially thought that this DNA might have been junk, that was actually called junk DNA for a lot of the 90s. So this DNA that didn't code for anything. And they thought it might be a bit like a fossil from our genome, a bit like our appendix. Like our appendix is there. We used it once, but we don't anymore, and it's pretty non-functional now. We don't need our appendix. We can easily have it removed, and it doesn't affect our day-to-day life. But DNA that's in the human genome, there are some fragments that you could do. If you lost it, it wouldn't really matter, but not that much. So there's a lot of DNA that doesn't code for stuff, but is really important. And actually, it was shown that when you look at a gene, it's not just the bit that codes for the protein that's important. It's all of the bit that surrounds it as well. And it was shown that in 2005, using computational work and bioinformatics, they showed that 43% of all genes contained a sequence that could fold into one of these cube-like structures. In 2013, it was proved that these could exist in cells using special antibodies that could be lit up using fluorescence. And this was even in BBC News. So these types of special DNA structures, or these quadruple helices, exist in human cells, and they exist in humans. And well, what do they do, and why might they be important? So we know that DNA exists as a double helix, but it could also exist in these special DNA shapes. So these ones here are called G quadruplexes because they form in sequences that have got a lot of the base G. They're quadruplex because they have 4 strands. And these are called I motifs. And they're called I motifs because they are intercalated, which means that they're like your interlocking fingers. And these special DNA shapes actually have a role. They have a purpose. And that's the reason why some of our DNA might not code for something, but the shapes that it could form help guide whether a gene is switched on or off. So if we have our DNA in our normal ladder form, our double helical conformation, we would get a normal amount of the gene. But if we encourage the DNA to form these special shapes, this can help determine whether we have a gene switched on or whether we have it switched off. So what does that mean in terms of us reading the genetic code? So let's say we want to think about how we might switch on a gene. And it's not binary. It's not like a light bulb. You can either have it on or off. It's usually that you would switch it on a little bit more. So think more about one of those switches where you can actually adjust the amount of light that you're getting. So rather than on off, it's more like you can turn it up and you can turn it down. So let's go back to our cakes analogy. If we've got a bit of DNA and that gene is being switched on more, what's happening is we're making lots more of RNA. And then those copies are then making lots more protein. And I would liken it to if you were using your Mary Berry 100 Cakes and Bakes and you wanted to bake some cakes for a fundraising event at one of your Diabetes UK support groups events, you might take your recipe for your cupcakes, make a whole load of photocopies or working copies of that recipe, give them to your committee members, and then you can go and make lots and lots of cakes. And that's essentially what's happening when we're switching on a gene. We make lots of working copies that's then used to make lots of protein, but the amount of DNA remains exactly the same. So diabetes, we know that genetics plays a role in diabetes, and diabetes is actually mostly genetic. There are some non-genetic parts to it, and there are some things that you can do to affect the genes. There's always something that you potentially can do, but the majority of it is genetic. And the gene for insulin has a special polymorphic region. And the reason it's special is because it changes in length and also slightly in sequence between different people. And that's why they call it poly, so many, morphic, so many differences. These polymorphic regions are actually, they're all across the human genome. And it's these regions that are used in DNA fingerprinting. So the stuff that's used for crime scene analysis. And the reason they're used in DNA fingerprinting is because everybody's is individual. So in the gene for insulin, this polymorphic region is repeats of these sequences. And they will repeat again and again and again and again and again. Now you will notice that one of the strands has lots of guanines and the other strand has lots of cytosines. So this part of DNA can form these special structures. So you can form the G quadruplex, or you can form the I motif rather than lots of ropey double helix that I've just drawn there, but you get the picture hopefully. So this is the insulin gene. And this region here is the ILPR, the insulin-linked polymorphic region. And you can kind of see it's a really nice, actually, it's a really nice repetitive set of G. And you can kind of see that pattern. So that's the region of DNA that we're interested in. So before we started this project, we already knew that this region of the insulin gene was very special. And we already knew that it could fold into these unusual DNA structures. And we knew that this could affect how much insulin is made and potentially have a link with the development of diabetes. But we didn't know, we couldn't really tell much more than that. We also, funded by Diabetes UK, previously to this current funding that we have, had found some compounds that could switch on the insulin gene. So we do experiments in cells that we grow, and we found that we screened a lot of compounds, and this compound here is as good as glucose at enabling switching on or starting producing insulin in these cells. And you'll notice actually we've also got, we did have a compound that could do the complete opposite, though it's obviously not something that we were interested in developing further for a drug, if that makes sense. Don't want to be switching it off. We would like to switch the insulin gene on rather than off. So the aims for this current project that we're currently funded for is, could we improve the compounds that we've already made so we could use them as tools and learn more about why this happens? What do the compounds actually do? What proteins bind these DNA structures? And then also, how does genetic variation and genetic mutation affect these structures? And the potential impact of this is these compounds that we've found, these could be leads for potential treatments, new treatments for diabetes. They're definitely tools that we can use to learn more about how diabetes develops and how this special region of DNA could be triggered or affected. And it could be that we might be able to then predict types of people that might get diabetes. So with these compounds, we had probably about, I think it was about five or six compound classes that we had at the start of this project. And we had a researcher that synthesized over 200 new compounds. So these were quite similar to the ones that we'd already found, but they have different properties, and it enables us to just see which one of them is best. And these are what we'd call drug-like, so they could be developed into a medicine in the future. And we use a type of cells called INS1 cells, and this is what they look like here. They're rat cells, and they're seeded in flasks. So we put 2 million in per flask, and this is basically times 10 object magnification, so you get an idea of how big they are. So they're tiny, but we grow them in our lab, and that's what we do our experiments on. So because rats have different DNA to humans, what we do is we add some special DNA, and we add a segment of DNA called a plasmid, and this has human DNA in it. And we also add a control, and we use 96 well plates. So this is what they look like here. So each one of these different wells, we can do different experiments in them. And before we do anything with our cells, we starve them overnight. So it's a bit like doing a glucose tolerance test. We starve them overnight from glucose, and when that happens, their morphology changes, and we can see that. And then we can treat these cells with glucose, and we use that as a positive control. So we treat them with glucose, and then if the cells are behaving normally, they will start expressing insulin. So you'll see insulin go up. And what we also do is then we compare the compounds that we found. So for example, after 1 hour of being treated with glucose, we see a big increase in the production, or the production of that gene. And then we see the same thing with this compound. And the more compound we use, the higher the effect that we see. And something else that we found is that we looked at different variants in the sequence. And the ones that form the special structure respond to glucose, and the ones that don't form special structures do not respond to glucose. And this is something that we're looking into more deeply, and we're interested in greatly, because this could potentially enable us to look at somebody's sequence using DNA sequencing and be able to potentially predict the behavior and how their DNA is going to affect how their insulin is read. And something that we're developing now, because we've just coming towards the end of our Diabetes UK project, we're actually starting a new project which is funded by the government, which is the BBSRC, the Biotechnology and Biological Sciences Research Council. And what we're looking at is, what is the relationship between the sequence of the DNA, the structure that it forms, the function in the genes, where these form in the genome, and how that changes with age. And one of the example genes that we're looking at is the insulin gene. So this is building on the work that we've already done with the project that's currently funded by Diabetes UK. So I'll stop here and allow you to ask any questions that you might have, but I need to acknowledge the people that do the work and the people that have been working in a team doing this. So this work was done by Dr. Rupesh Shikale and Dr. Dilek Ganeri, who is on the call today. And then this is the rest of the research group who are all interested in DNA structures and how changing their shape of DNA can affect different diseases and different the ways that we can apply it in biology. So thank you very much for your kind attention, and I'll be happy to answer any questions.
Thank you so much, Zoe. Thank you very much. Fascinating talk. I certainly have learned a lot, Zoe, over the past half an hour. So I'm very grateful. Let's see if we have any questions from the group.
Hi Zoe, it's Nyla.
Hi,
I was really
Nyla.
interested because I'm type 2 diabetic, and when I speak to my nurse, they're not sure whether my diabetes is getting worse because it's genetic, or that they need to increase my metformin, which I'm trying to say I don't want any more metformin. And they said I haven't done enough research yet to know whether I'm going to get worse because it's genetics, or whether I just need to change my diet or take more medicine. So this is really interesting for me, but how far away are we, do you think, from getting any, medication that could change how it affects me or, anyone like myself?
So there's some things that really need to sort of be clear before we kind of are able to then decide, like, on an individual basis, why, what's the best thing for an individual person. And this personalized medicine is really where the NHS was really going for just pre-COVID. So there was a plan to do whole genome sequencing for all, and COVID really derailed that. And I'm really sad to see that the NHS as it currently stands is in a bit of a state, and needs to be resolved before we start looking at everybody's DNA, if that makes sense. But they're still, Genomics England are still going through and doing different types of people, so people with rare genetic conditions, but also people with cancer. So I think they have well over 100,000 genomes that we can use and look at as researchers. And one of the things that I would like to do, and this would be separately, I'd have to get separate funding to do this, is to look at actually those people in those from that population, to be able to then see, well, we know the sequences that fold into the special structures. How many of those people that have diabetes, what types of sequences do they have? Do they fold into these weird structures, or do they just keep to the normal structure? What is the difference between those types of people? So potentially what I could see coming out quickly from that was maybe a prognostic. So looking at those sequences, once we've got that information, being able to predict, because that could then help people that, perhaps you might be predisposed, you might then be able to be careful with lifestyle changes, or you might be able just to look out for symptoms, because for some conditions that, if you get diagnosed quickly, you can have some treatments to reverse it. And the quicker you kind of know that you've been triggered, the quicker you can do something to reverse it. And in cases like yours with type 2 diabetes, there will be, I don't think it's a case of there's just type 1, there's type 2. I think that there's a whole grade, there's a gradient of different types of diabetes. And I think that's been acknowledged now, but how to deal with that and how best to move forward with that is something that we need to do a lot more work to do. So it might be that diet and exercise could be really beneficial to you, but it could be actually that, no, you just have to have more medication. But being able to have a better understanding of that means that you won't be in so much limbo So you wouldn't be like, well, diet and exercise might help me, but I don't know. So you might do it and you're like, well, because I mean, as we've just heard, changing your diet's a really big thing. Like, and it's one of those things that it's like a freedom you don't want to have to sort of give up. So there is a lot to kind of unpack there. And it's not just, so we're only looking at one aspect of this. We're just looking at the insulin gene. There are other genes that will play a role in it as well. So it's sort of multifactorial. So what I'm trying to sort of explain is what the easiest things that we could do are, but also biology is complicated, human beings are complicated, and individuals are complicated. But I do think we've got a massive opportunity here with the work that's already been done to be able to, because their genomes are connected to NHS digital data, so we can do that. But we need to get somebody to do it, pay someone to do it. So yeah.
Well, thank you very much, Zoe.
You're welcome.
I think we have Lindsay with a virtual hands up.
Yeah, hi there. Really, really interesting on two levels for me. I firsthand I've been on the receiving end of having a genome mapped when I was diagnosed with breast cancer 6 years ago. And I was lucky enough to be under the Marsden, and they were able to map the genome of my tumor, which directly decided the path of treatment I could take. Now that's outside of genetic testing for BRCA genes and all of this.
Yeah.
Luckily I didn't have the BRCA gene, but it was called the Oncotype DX, which I'm sure you've heard of. And that made me able to decide that I didn't need to have chemotherapy. It informed my decision to stop taking tamoxifen after 5 years, comfortably knowing that I'm not going to have a recurrence. And so what you're describing for diabetes, it's there, it's ready to be done, like you said. And then interestingly, I got diagnosed out of the blue with type 1 3.5 years ago. And I have no family history of type 1 nor type 2. So we may never know what caused it, but maybe down the line, my genome will be mapped and somebody will be able to say, okay, this is what's kick-started it, and which I am now the family history that's starting. So my daughter, who's now 7.5, I've had her on the trial net. So, I'm pretty confident she's going to be okay. But the work that you're doing is probably going to diagnose her children or not, as the case may be, or prevent. And I just think it's absolutely fantastic. And having been on the receiving end of it myself, and probably saved me a horrible chemo journey, and then my grandchildren will probably then be on the receiving end of it from the insulin and diabetes point of view. It's fantastic. It's absolutely fantastic. Sorry, my cat is howling in the background. She's 18 and senile. Sorry.
Oh. So I think that what you've just described is is the way forward for a lot of medicine, and it not just diabetes. Like if we can, that personalized healthcare is going to be so much better for people. Like I think most people will have had some experience of taking a medicine and it perhaps not working for them, or of hearing someone saying, well, this medicine, might work for you and it might not. Being able to do genetic testing and being able to say, actually, you don't have the right enzyme to break this down. It's not going to work, or we're going to have to give so much of it to you that you're going to have awful side effects. Having a better understanding is going to improve our healthcare all round. But what we would like to do for diabetes is to be able to kind of discern, like, what is the trigger point? Because some of the things that we have found with these structures is that you need only like a couple of bases to be different. So only a couple of letters need to be different for it to go from one structure to another. And I've worked in this area for a number of years, since 2005, and I was astonished at that. I thought there would need to be much more of a change, but that level of change could be triggered by somebody having a cold or somebody getting ill. So it could be that we're looking for a trigger point, that somebody might actually have, like, be one mutation away from being triggered to have diabetes. You know, this is our current working hypothesis for this particular area. And that's what we're going to be looking in for this next project, which is funded by the BBSRC, is looking at how, like, what's the tipping point before you go from the double helix to the special structure. And then how does that change with aging? Because we get a lot more diseases and conditions as we age. So that's what we're going to be looking at with that particular one. But I will be applying for further funding to investigate what we found out more, because we opened so many cans of worms that now we're like, well, this is really interesting, but where do we go now? So, but that's a process that takes a long time, is to apply for the funding. We've got enough preliminary data, apply for the funding, get the funding, do the work. That's the bit that takes the longest. If we didn't have to do that, this could be answered in a couple of years, but that's the bit that takes the longest.
I think it's, yeah, I think it's also hard for people to be able to put it into context when they don't see it working and it's still at the research phase.
Yeah.
Like, what I really want to put across to everyone here is that it does work, and it does matter, and it does affect people. And unfortunately, diabetes is down on the list of priorities behind cancer research, and you know, that's the way it is. But it's if you're working on it and you're getting there, it works, and it can, like you said, it's tailor-made medicine at the end of the road, which can only be beneficial for the system, for those of us who need it, for like my mother-in-law is type 2, and she's on I don't know how many different medications, and it's just like, oh, let's try this one and see if it fits. And the amount of different things she has to go through, and the symptoms that, or the side effects of these medications, whereas in 20 years time someone might say, right, we've mapped this is all you need, and you know, off you go. Yeah, it'd be brilliant.
Thank you for your insights, Lindsay. I think that's really helpful for, yeah, because I can explain it the best, you know, as best I can, but to have somebody that's kind of seen it firsthand and actually be like, this is possible, that's actually really helpful. Thank you.
Thanks.
Can I jump in? Oh yeah, please go ahead.
Sorry, does this mean that, so for type 1s, it would be possible to know that they've got a predisposition to become type 1? Or are you saying that maybe something has to happen for the bit to make?
There will, I'm pretty confident. So my hypothesis is that, yeah, you'll be able to predict, and that actually people with type 1, they usually have like a trigger event. It might be that they have a bereavement, or they have a very stressful situation, or they get chickenpox or something, or, you know, or they've taken a medication. There's usually something that triggers type 1 diabetes, and it's that triggering of our immune system to attack the beta cells. That's, you know, we want to kind of work out, well, why, you know, what's the trigger point? And there's evidence to show that already in the literature, stuff that people have already worked on, that this polymorphic region, some are predisposed to being like to trigger type 1. And even it's not really clear to me now, even knowing what I know, why that would be the case, but it's definitely something that we want to explore further. Because it doesn't, when you say, oh, type 1 diabetes, people know it's an immune condition, you know, it's your immune system sort of destroying your capability of making insulin. So why would it be in the polymorphic region? That's, you know, but we should be able to quite easily work out whether or not that's the case. but yeah, so I think you're right there.
If I can just come in, so a couple of things. One of the things that we sort of consider, and for instance, in a more recent research locally in Sutton as well, very often there seems to be a difference between races in terms of prevalence of diabetes in the population. So my question would be, through the research specific to around DNA, do you see any differences there as well?
We've not looked at yet at the different genomes. It's something we want to do, but again, that's a categorization that would potentially easily be able to do, like be able to kind of, but it might be, so there are different things that you could unpick there, but you'd need to look at the information, like the data, but different races like will have different genetics that are, like if you're from a different race, your genetics are going to be sort of similar. So it might be that, it's just that, or it might be a cultural thing. But what they've shown is that it's probably a mixture of two things. When I say cultural thing, like differences in
lifestyles,
lifestyles,
exactly.
but it is likely to be both. But we would really like to be able to predict that, for testing. The NHS was going to be testing newborns. That was their plan before COVID, to test all newborns. And as a parent to a baby, they do a lot of genetic testing anyway, but to be able to kind of be looking out for, okay, you have a predisposition for this.
Yeah.
We can be looking out for symptoms. And then if we do, we can get testing very quickly, rather than going through months of, well, this is a bit weird, and what's this, and then hanging around and waiting and being referred and losing valuable time to have, there's like immunotherapies that can be provided now to sort of reverse things.
Yeah, let alone the economical advantages as well of actually doing that at the beginning of one's life. I think there is an intervention from Martin in the chat.
Yeah. So, yeah. My mum thought I got diabetes after German measles, and that's really common. Lots of people, they have a trigger event, get a condition. And as somebody that works on DNA, we know that these conditions, each time you have a condition or you have an illness, your immune system changes. So everything you get exposed to, your immune system changes slightly. And it does mean that you can acquire an allergy at any point in your life. And it's like, well, I'm not allergic to that. I'm like, you could have got a cold and also acquired an allergy just after getting a cold. So these things are interrelated, and we need to learn more about it to kind of know, well, what's the critical point that then triggers this and triggers the diabetes.
The other thing that I wanted to ask, very often in sort of talks, and our guests as well talk about the importance of diet, importance of exercising. I think what you said earlier is that yes, that might play a role, but actually mainly the path to diabetes is predefined through the DNA. If you had to put sort of percentage on it, I'm not sure if it is possible at all, but if you had to, what would you say? To which extent actually it is the DNA, and to which extent can we just sort of do a little bit of work around the edges to mitigate?
So I think there's always something you can do to mitigate. That's my opinion. That I think there'll be people that say, oh, well, it's just genetics. There's no point. But actually understanding your genetics means that you should have more control. Like, there are people that can eat a poor diet and they're not going to get diabetes. Lucky them, but there are some people that if they have diabetes, they could potentially control with diet. Actually, it's not going to be easy, but they could control it with diet. And certainly I do have a little bit of experience of that with myself, because their diabetes is in my family history. So both my parents have had diabetes. My brother has diabetes. My aunt has diabetes. My grandparents had diabetes. In fact, I'm the only person that doesn't currently have diabetes. But I got gestational diabetes when I was pregnant with my first son. And there what did seem to be a very strong relationship between exercise, not even diet, just exercise, and the level of sugar in my urine. And to the extent that when I got pregnant the second time, I was like, right, well, I'm not going to stop exercising. So I cycled to work every day. And the only times they found sugar in my urine was when I went on a conference. Well, the two times I went on conferences and I wasn't cycling to work. And I was actually involved in a clinical trial with my last I'm just permanently pregnant. With my last pregnancy, I was involved in the clinical trial, and I was, I was like very, very explicit. I am doing exercise every day, and even when I'm not cycling to work, I'm on my exercise bike to make sure that I can still eat cake. Because it's really like controlling. Because I think with my first pregnancy, controlling my blood sugar with diet was really difficult, actually, really, really difficult. And I couldn't, like, you'd look at the NHS website for their advice. It was like, eat some toast with some jam. I was like, what? I couldn't get away with like even the thinnest slice of toast with all roughage, the multi-grain. I couldn't tolerate any of that. No, so I was basically just eating protein instead. But yeah, so I do think that there is, on a personal level, I think I'm predisposed to getting diabetes. I know that. And it's, for me, it's a matter of just how long I can carry on my normal life without finally getting it. So that's why I think, well, for me, like if I keep exercising, I should be okay. But I would like to be other people to have a bit of a better idea of, I've kind of worked that out for me, but other people to be able to just do a test and say, actually, if you do this, this, and this, you might not have to take medication. Some people want to, they're quite happy taking medications, but some people aren't. And we should give people the opportunity to be able to take control themselves of their life.
Absolutely.
So,
wonderful. Have we got any final questions to Zoe from the group? That silence might tell us no. I think everybody is happy. So that leaves me just to say, Zoe, a big thank you on behalf of the group as well. Thank you for your time. You obviously are a big friend and supporter of Diabetes UK, and it's just such a great pleasure to have you in a sort of more intimate circle of our group as well this evening. So thank you very much.